Hi everyone. Today I am going to start the new chapter Cell, the Unit of Life. Under this I will explain definition of cell, scientists' contributions, cell theory, cell morphology, sizes of cell, shapes of cell and prokaryotic cell structure. Very first we have to know the definition of cell. All organisms are composed of cells. Some are composed of single cell and are called unicellular organisms, while others like us composed of many cells are called multicellular organisms. Here every living organism composed with cells and some of them are composed with single cell called unicellular organisms and some of them composed with many cells which are called multicellular organisms. Unicellular organisms are capable of independent existence and performing the essential function of life all or occurs in single cell. Anything less than a complete structure of a cell does not ensuring independent living. Hence, cell is the fundamental, structural and functional unit of all living organisms. Now, historical events in cell biology. First cell discovered by the scientist Robert Hooke in cork tissue of oak tree or Quercus sober. Anthony van Leeuwenhoek first saw and described a living cell. Robert Brown later discovered the nucleus in a orchid plant. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. We know Robert Hooke is the first observer of cell but in non-living state. Anthony von Leeuwenhoek is the first observer of cell in living state. Robert Brown is the scientist who observed nucleus for the first time in orchid plant. The invention of the microscope and its improvement leading to make the electron microscope revealed all the structural details of cell. Cell theory. In 1838, Matthias Sleden is a German botanist examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, the scientist Theodore Swan in 1839, his British zoologist, studied different types of animal cells and reported that cells had a thin outer layer known as plasma membrane. He also concluded, based on his studies on plant tissues, that presence of cell wall is unique character of the plant cells. Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Sleden studied on large number of plants. Finally, he concluded plant body composed of different kinds of cells in the form of different tissues. Theodore Swan, he studied on animal cells. Finally, he reported animal cells covered by plasma membrane or cell membrane without cell wall. Matthias Sleden and Theodore Swan both together and formulated a theory called cell theory, which is also called cell doctrine. This theory did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained that cells divided and new cells are formed from pre-existing cells, which means omnis cellula e cellula. He modified the hypothesis of Sleden and Swan to give the cell theory a final shape. According to this, all living organisms are composed of cells and cell products, and all new cells arises from pre-existing cells, which means omnicellula e cellula. Sleden and Swan together formulated the cell theory or cell doctrine. But this theory did not explain how new cells were formed. Rudolf Virchow first explained that cells divided and new cells are formed from pre-existing cells. He given the statement in Greek language called omnis cellula e cellula. Rudolf Virchow modified the hypothesis of cell theory and gives a final shape. According to this, all living organisms are composed of cells and products of cells. All new cells are arises from pre-existing cells. An overview of cell. The onion cell, which is typical plant cell, has a distinct cell wall as its outer boundary, and just within it is cell membrane. Cells that have membrane bound nuclei are called eukaryotic cells, whereas cells that lack a membrane bound nucleus are called prokaryotic cells. In both prokaryotic and eukaryotic cells, a semi fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main area or zone of cellular activities in both the plant and animal cells. Various chemical reactions occurs in it to keep the cell in the living state. A typical plant cell contains cell wall as outer boundary. Just within that they contains cell membrane or plasma membrane. The cells which contains membrane bound nuclei are called eukaryotic cells, where the cells which lack of membrane bound nucleus are called prokaryotic cells. Here in both prokaryotic and eukaryotic cells contains a semi-fluid matrix or liquidous living matter is called cytoplasm, which occupies the volume of the cell. Cytoplasm is the main area or main zone for cellular activities, and various chemical reactions occurs in the cytoplasm to make it as living state. Besides the nucleus, the eukaryotic cells have other membrane bound distinct structures called cell organelles like endoplasmic reticulum ER, Golgi complex, lysosomes, mitochondria, microbodies, etc. The prokaryotic cells lack such membrane bound cell organelles. Ribosomes are non-membrane bound cell organelles which are found in both eukaryotic cells as well as in prokaryotic cells. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles like chloroplasts in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centriole, which plays very important role in cell division. Not only nucleus, eukaryotic cells have other membrane bound cell organelles also like endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, etc. But in prokaryotic cells, there is no membrane bound organelles. Ribosomes are the cell organelles which are non membrane bound organelles found in all cells, both eukaryotic cells as well as in prokaryotic cells. Ribosomes are the cell organelles not only found in cytoplasm, also present in some cell organelles like chloroplasts, mitochondria, and rough endoplasmic reticulum. Animal cells contains another type of non-membrane bound cell organelle called centrioles. Centrioles which helps in cell division, particularly in the formation of spindle fibers. Size of cell. Cell differ greatly in size, shape and activities. Mycoplasma is the smallest cells, only 0.3 micrometers in length. Bacteria, the size of bacteria is 2 to 5 micrometers. Largest isolated single cell is egg of an ostrich. Human red blood cells up to 7 micrometers in diameter. Nerve cell is longest cell. which is up to 100 microns. Cells are greatly differs in their sizes, shapes and cellular activities. Mycoplasma is the smallest cell, which is only in 0.3 micrometers. Bacterial size is 2 to 5 micrometers approximately. Largest isolated single cell is egg of an ostrich, which is 12 to 15 inches. Human red blood cells up to 7 micrometers in diameter. Nerve cell is longest cell, which is up to 100 microns. Next, shape of cell. The shape of the cell may vary with the function which they are performing. Based on this, cells may be disc-like or polygonal or columnar or cuboid or thread-like or even irregular shapes. Let's see the diagrammatic representation of various cells in various shapes. Very first one, red blood cells RBC, round and biconcave shape. White blood cells, which are in amoeboid shape. Columnar epithelial cells, long and narrow shape. Nerve cells, branched and long. A trachea is long elongated cell. Mesophilic cells are round oval shaped cells. Remember, RBC in round and biconcave shape, white blood cells in amoeboid shape, columnar epithelial cells in long and narrow shape, nerve cells in branched and long, tracheids in long elongated shape, mesophilic cells are in round and oval shape. The next topic is prokaryotic cell and its structure. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma or PPLO, which means pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than eukaryotic cells. Prokaryotic cells may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, which are in rod shape, coccus, which are in spherical shape, vibrio, which are in comma shape, and spirillum, which are in spiral shape. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibits a wide variety of shapes and functions. Bacteria, blue-green algae or cyanobacteria, mycoplasma or PPLO are best examples for prokaryotic cells. Generally, prokaryotic cells undergoes rapid cell division than eukaryotic cells, and prokaryotic cells shows great variation in their shapes and sizes also. For example, bacillus bacteria, which are in rod shape, coccus bacteria, which are in spherical shape, vibrio bacteria, which are in comma shape, spirillum bacteria, which are in spiral shape. The organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is called cytoplasm. There is no well-defined nucleus in prokaryotes. The genetic material is basically nagged, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA is present in bacteria, which is called plasmid. Many bacteria have small circular DNA outside the genomic DNA. These smaller DNAs are called plasmids. See, all prokaryotic cells having cell wall except mycoplasma, and all contain cytoplasm, and they does not contain well defined nucleus. Genetic material of prokaryote cells is basically nagged, not enveloped by any nuclear membrane. Prokaryotic cell. In addition to the genomic DNA, Bacteria have small circular DNA which is called plasmid. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In next chapters, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. Plasmid is the unique feature of bacteria. It gives antibiotic resistance to bacteria. No organelles are like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotic cells having unique form of inclusions and cell organelles. Cell envelope and its modifications. Most of the prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, which means outermost glycocalyx followed by cell wall and then the plasma membrane. Although each layer of envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two different groups on the basis of the differences in cell envelopes and the manner in which they respond to the staining procedure developed by the scientist Christian Gram. The prokaryotic cells, particularly the bacterial cells, have chemically complex cell envelope, which is composed of glycocalyx followed by cell wall and then plasma membrane or cell membrane. Each layer of cell envelope performs distinct function and they act together as a single protective unit. Bacteria can be classified into two different groups based on how they are responding to the staining procedure developed by the scientist Christian Gram. The bacteria that which can take up the gram stain are gram positive and the others that do not called gram negative. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting and collapsing. The plasma membrane is selectively permeable membrane and it interacts with the outside world. The membrane is similar structurally to that of eukaryotes. Remember, the bacteria which can take up the gram stain called gram positive bacteria, which do not take up the gram stain called gram negative bacteria. Glycocalyx differs in composition and thickness. In some bacteria, it is loose sheath called slime layer. In some bacteria, it is thick, tight, and tough, which is called capsule. The cell wall determines the shape of cell and provides strong structural support to prevent the bacteria from bursting and collapsing. Plasma membrane is selectively permeable membrane. This membrane is similar structurally to the plasma membrane of eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, distribution to daughter cells. They also help in respiration, secretion process to increase the surface area of plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into cytoplasm called chromatophores, which contains photosynthetic pigments. Special membranous structures called mesosomes, which is formed by the extensions of plasma membrane into the cell. A special membranous structures is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules and lamellae. Mesosomes helps in cell wall formation, DNA replication and distribution to daughter cells. Mesosomes also helps in respiration, secretion process to increase the surface area of plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions which are called chromatophores, which contains photosynthetic pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts such as filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides the flagella, Pili and Fimbriae are also structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of special protein. Bacterial cells may be motile or non-motile. If motile, they contains filamentous extensions called flagella from cell wall. Bacteria shows a range in the number and arrangement of flagella. Flagella is composed of three parts: filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, bacterial cells also contains pili and fimbriae. The pili are elongated tubular structures made up of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Fimbriae are small bristle-like fibers in bacteria which helps in attachment. Ribosomes. In prokaryotes, ribosomes are associated with plasma membrane of the cell. They are about 50 nanometers to 20 nanometers in size and are made up of two subunits called 50S subunit and 30S subunit, which when present together form 70S prokaryotic ribosomes. In prokaryotes, ribosomes are about 50 nanometer to 20 nanometer in size and contains 70S type of ribosomes, which are composed of 50S and 30S subunits. Ribosomes are specially the sites of protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate mRNA into proteins. Ribosomes are the sites of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes or ergosomes. These polysomes helps in the translation of mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Examples: phosphate granules, cyanophician granules and glycogen granules. The reserve food material in prokaryotic cells are stored in cytoplasm in the form of inclusion bodies. These inclusion bodies are non-membrane bound organelles. They are freely lies in cytoplasm. Example: phosphate granules, cyanophician granules and glycogen granules. Gas vacuoles. Gas vacuoles are generally found in blue-green, purple and green photosynthetic bacteria. Generally, gas vacuoles are permeable to gases and impermeable to liquids. Gas vacuoles helps in osmoregulation and buoyancy. Gas vacuoles are generally found in blue-green bacteria, purple bacteria and green photosynthetic bacteria. Generally, these are permeable to gases and impermeable to liquids. Gas vacuoles generally helps in osmoregulation and buoyancy. Let's check the diagrammatic representation of prokaryotic cell, especially bacterial cell. Let's see how they contains capsule, cell wall, cell membrane or plasma membrane, cytoplasm, inclusion bodies, ribosomes, genomic DNA, plasmid DNA, flagella. Let's see this diagram carefully for your better understanding. Up to here, we completed cell, the unit of life, introduction, scientist contributions, cell morphology, sizes and shapes, and prokaryotic cell structure. In next video, I will explain about eukaryotic cell structure. Thank you.